Okay. Okay. So first of all, I really want to thank the organizers and everyone for inviting me. And I'm really excited to be here. So I've only been here for today, but really, really exciting talks. And thank you so much for Julia for everything she's doing. I'm going to switch gear and I'm going to talk about microbiology. And so I know that a lot of you are not microbiologists, so please stop me if you're not sure of what I'm saying, if you're not comfortable or you have questions. So it would be much easier than me just chagging along and you not understanding what I'm saying. So I'm going to talk about how we navigate the ripple effects of wastewater irrigation on soil quality, crop health, environmental sustainability. And I'm just going to start by showing you where we're located. So you were showing all these beautiful photos, and we're located in the desert. And because we're located in the desert, and Israel is a desert country, then we have to use wastewater, or we have to use it for irrigation. Using potable water is not really an option. And so what we're doing is we're using wastewater for irrigation. It's been being used for over 50 years now. This is not the only use for wastewater, of course, and in California, they're mainly using it for drinking water. So they're desalinating wastewater to drinking water, and that's the main use of the water. But in Israel, many, many years ago, it was decided to use it. Currently, around, and Ilya will correct me if I'm wrong, it's anywhere between 80 and 90% of the water is being used for irrigation. All the fruit trees in Israel are irrigated with treated wastewater forever. Okay, so for at least 40 to 50 years. And this is where we're located. So this is the Water Institute. I belong to the Water Institute. We're doing desalination, hydrology, microbiology. And so we're interested in the wastewater irrigation. We're interested in different aspects of wastewater irrigation, but I'm going to talk today, or going to mainly talk about contaminants that are related to wastewater irrigation, to treated waste. Of course, treated wastewater, nobody's using wastewater for irrigation, but treated wastewater for irrigation. And I'm specifically talking about secondary treated wastewater. So in Israel, we're also having, so we have the natural water, we're using more and more, desalinated water, and I'm just gonna, okay. So we're using, where is it? Has to be somewhere. I can see it. So we're mainly using treated wastewater for irrigation, and we're desalinating a lot of the water. So over 70% of the water we're drinking is desalinated water. And so we're using more and more water for irrigation. Now it's kind of tapered down, so it's kind of leveling, but we're using more and more treated wastewater for irrigation. And so it's either tertiary treated wastewater or secondary treated wastewater. Secondary treated wastewater are used for irrigation, are restricted. Okay, so that means that they're mostly used for fruit trees and for a non-edible crops. Well, tertiary treated wastewater are used for unrestricted irrigation. So what's tertiary treated wastewater? That's open for interpretation, because secondary treated wastewater is usually activated sludge, some kind of biological treatment. Sorry.
Tertiary treatment is also, right?
No. Tertiary treated could be anywhere between soil filtration to desalination. The whole range is basically called tertiary treatment. So you can, actually you can name it. Okay, so you can say, okay, I treated my water with soil filtration, and that's tertiary treatment. But it also can be, it could be desalinated to a potable water level, and this is also tertiary treatment. So it's a huge range, and so we are using secondary treated, as I said, for irrigation of non-edible crops and for fruit trees. And we're also using tertiary treated wastewater which are basically secondary treated wastewater that are discharged into the aquifer and then pumped south of where they're infiltrated into the aquifer, and then they're using for unrestricted irrigation. So our question was, okay, so if we're using secondary treated wastewater, we know that in the secondary treated wastewater, we have pathogens, different type of bacteria, different type of viruses, also pathogenic viruses, protists, all kinds of pathogenic protists, and antibiotics. Okay, and a range of other things, microplastics, other types of contaminants, but I'm going to focus in this talk only on these contaminants, or so the pathogens, bacteria, viruses, and protists, and on antibiotics. But just to keep in mind that there are other contaminants, and we're also thinking about these other contaminants. So, sorry. So our objective was, well, if you have so much secondary treated wastewater, and we're also bordering countries like Jordan and Egypt and other countries like Lebanon that do not have tertiary treatment. And so could we use the secondary treated wastewater and expand their use beyond the use of restricted irrigation? That was our initial question. And this was a project we did with the Palestinian Authority, where we were trying to think, maybe we can use the secondary treated wastewater, not to bring it to tertiary level, but maybe we can use it for unrestricted irrigation. And so what we wanted to do is to evaluate the health-related impacts of using secondary treated wastewater when we're irrigating vegetables. Okay, so this is basically unrestricted irrigation. So the first thing we did, we had we established a study where we irrigated tomatoes with 3 types of water: potable water, so water from the tap, secondary treated wastewater, and we used two types of secondary treated wastewater. So we used more heavily treated wastewater, it's the secondary treated wastewater, so chlorinated and non-chlorinated secondary treated wastewater, and then tertiary treated wastewater. And this tertiary treatment was basically sand filtration, which is, again, is considered tertiary treatment. And so this is Ezra, and Ezra was a PhD student, and he was doing most of this work. And we're doing it together with UC Davis. And what we did was, okay, so we evaluated some indicators, not pathogens, but indicators in the water. And the x-axis is the different indicators, and really the names don't matter, but just different indicators. The y-axis is the concentration of the indicators per 100 ml. And then the 2 lines you can see here are the WHO standard and the Israel standard. And because the Israel standard are equal to the EU standard and the standards of the EPA, and the WHO standards are higher because they want to accommodate different countries, okay? But you can see that the quality of our water was very, very low, okay? So it was higher, basically, than the high standards of the WHO, okay? So the water was pretty bad. Not all the water, you can see the different colors. So it's secondary treated and tertiary treated. And this is over two years where we are sampling the water every couple of months. And then so we looked at the soil. So we tried to see how it affects the soil, again, over the 2-year experiment, soil experiment. So it's all done in the field. And when we looked at it, so again, you can see the different colors. The x-axis is, again, the different indicators, and the y-axis is the concentration of bacteria. We were looking, and nothing. Okay, so it doesn't matter what type water we use, what quality water we use, we didn't see any difference. And when we looked at the tomato wash, so we washed the tomatoes, and this is normalized per tomato. This is the soil is normalized per gram of soil, and this is normalized per tomato. And so we normalize this per tomato. And again, treated wastewater, secondary and tertiary treated wastewater, potable water, we look at the tomato wash, doesn't matter. Whatever we do, we find the same indicators or the same concentration of indicators, which was pretty interesting. Okay, so the next thing we did, and this is Mitiko and Rachel, and they did an experiment where they looked at antibiotic-resistant genes. And again, the antibiotic-resistant genes don't matter much, but this is what we found in the fresh water or the potable water. And then you can see what we find in the secondary treated, tertiary treated wastewater, what we find in the soil. And if you look at the tomato wash, there's no link between the water or what we find in the water, the antibiotic resistant genes we find in the water, what we find in the soil, what we find in the tomato, it was completely different. And so the reason, so we started digging into the, of course, at the beginning, I told the Rachel and Mitiko, I said, look, you probably made a mistake. I mean, you probably contaminated somehow, redo the whole thing. And they redid it, like, I think 3 times. And it eventually said, okay, so that's the result. And we started digging into the literature. And what we found is the tomato surface is very unique. We talked about it with Hunter earlier. The tomato surface is very, very unique. It's very hydrophobic. It's kind of binding a lot of bacteria. And so probably what we're seeing is basically dust. Okay, so whatever's in the dust, whatever bacteria in the dust, whatever antibiotic resistant bacteria are found in the dust, we're seeing it on the tomato, regardless of what's in the soil or what's in the water. And this is all drip irrigation. So there's no residual water on the tomatoes. And the soil is, and the tomatoes are not really close to the soil. Okay, so we don't think it's the soil. We don't think it's the water. So it's probably due to the tomato. Okay, so this is the results when we use different water, and we're saying, okay, let's use different types of soil. Maybe it's the soil. Maybe what we did was using a very specific type of soil, and then if we use different types of soil, and here you said a different concentration of sand and silt and clay, and so let's use different types of soil and cultivate something completely different. And this time we cultivated cucumbers and melons for over 2 years. And let's do the same thing. Irrigate with secondary treated wastewater, potable water, and maybe we'll see something different. And so we try to see whether we will find any differences. And again, we did find differences in the water, but we did find differences in the water, but we couldn't find any differences. And you can see again, non-irrigated water, potable water, treated wastewater. The concentration of the indicators was very, very similar, regardless of the type of water we were using in the different types of soil, clay, loam, and loamy sand. Okay, so we said, okay, so maybe what we're doing, maybe we need to treat the soil differently. Okay, so maybe what we need to do is just have different treatments of the soil, and then we'll see a difference. And so there are different types of soil treatments that are put in place when you're using treated wastewater. And so there needs to be barriers between the crop and the soil. And so barriers could be either plastic mulch or drip irrigation. Drip irrigation is another barrier, or subsurface drip irrigation. And so we did all the permutations. So plastic mulch, with drip irrigation, without drip irrigation, with subsurface, without subsurface, potable water, treated wastewater. We did everything. We were trying to see how it may affect. And these soil treatments are recommended by the WHO and the EPA and the EU and so different agencies. Okay, so the field setup, as I said, is all permutations of these treatments. And this is the field where we were cultivating, and again, we were cultivating that for 2 years. And so if we're looking at the water, then of course in the water we see higher concentration of indicators when we're using treated wastewater. That makes sense. Okay, it's lower than what you saw before because this is well-treated secondary wastewater. So well-treated, secondary treated wastewater. So the concentration of the E. coli and the enterococci and the fecal coliforms is within the range and below the range of the about WHO and within the range recommended in Israel and in the EU and by the EPA. And we were using 4 different indicators: E. coli, enterococci, fecal coliforms, and Pseudomonas aeruginosa. And then we were looking at the soil. And we were looking at the soil, again, whether we used potable water or treated wastewater, we didn't see any difference, nothing. So we were saying, okay, maybe we're just looking at the wrong thing. Maybe we're looking at indicators that have nothing to do with the pathogens that are found in the water, because there are huge number of pathogens and the indicators are only applicable and for, you know, a subset of the pathogens. And so we screened for 200 pathogens. So we did this huge survey where we were sequencing and looking for 200 pathogens within our soil, and within the water, the soil, the fruit surface, only the cucumbers, because nobody eats the surface of the melons, and then the puree of the cucumbers and the puree of the melons. Okay, so we mashed it, we surface sterilized it, and we mashed it. And I'm not getting into methods here, but I would love to talk to you about the methods, because methods are key to all these experiments. Okay, so how much water you're using, how much soil you're sampling, how do you do the fruit wash, and how do you do the vegetable puree? It's all, you know, it all took us a long time, and we had to estimate the limit of detection and the recovery, and so it's a lot of work gone into what you're seeing here. But to make a long story short, and if you're looking at this, and it doesn't matter, this is the subset of the pathogens that we actually were able to detect within those 200 pathogens that we were looking for. And if you're looking at the different matrices that we were using, then the overarching conclusion was that there's no link between the water, the soil, the cucumber wash, and the cucumber and melon puree. Okay, so whatever we put in the soil is meaningless. Okay, so we can irrigate to our house content. Now, bear in mind that if the concentration was very high, and the previous experiments, so when we started this project, the previous experiments were using very, very high concentration, the pot experiments with very high concentration of E. coli, and then you get contamination. Okay, if it's very, very high, then it's contaminated. But if you're using treated wastewater as recommended by all the agencies, then you're using the contaminants at the concentration that is meaningless to the soil. And so we were saying, okay, so maybe pathogens are not there, but maybe we can find something with the antibiotics and antibiotic resistance. And so what we were looking at a subset of antibiotics, and that was done together with Clinton. And then we were using the antibiotics and the antibiotic resistant genes that are related to these antibiotics. So we wanted to look at the link between the antibiotics that we may find in the water or the soil or the cucumber and the antibiotic resistant genes. So these are the different antibiotics that we were using, and this is what we were finding in the fresh, in the potable water and in the treated wastewater. And of course, in the treated wastewater, we find higher concentration of the antibiotics. Makes a lot of sense. But when we're looking at the soil, the only difference we were able to see is between the mulched soil and the non-mulched soil, and that regardless of the type of irrigation. And what we were seeing is that in mulched soil, we see an increase of certain antibiotics. Okay, so you can see here no mulch and mulch with specific antibiotics. This is tetracycline and afloxacin, that we see this increase while with other antibiotics like tetracycline and sulfamidazole, we couldn't see any differences. And then we kind of wrapped our heads around that. We're trying to think what's going on. And we found some papers showing that antibiotics like tetracycline, tetracycline and the afloxacin are actually binding to plastic mulch. And when they're binding to the plastic mulch, maybe what's going on is that they're binding to the plastic mulch and then they're released into the soil. And that's where we're seeing these increase in the antibiotics concentration. And but when we were looking at the antibiotic resistant genes, we couldn't see the link between the antibiotics that we're actually finding and the antibiotic resistant genes. And again, what you can see is that if you're looking at the water, the two types of water, they are not necessarily similar to what we're finding in the soil. And what we're finding on the surface of the cucumber is completely different, okay? And completely different from what we find in the tomatoes. But in the puree, in the inside the cucumber and melon, we found nothing, okay? Which is good news. But then we kind of thinking, okay, so maybe we have some heavy metals in the treated wastewater, and that's why we're having this increase in antibiotic resistance. And so we were trying to see, we're looking at zinc and manganese and iron, and trying to link between the different antibiotics, different heavy metals, and the antibiotic resistant genes, and nothing. Okay, we could not link between what we find in the water and what we're finding in the soil and in the puree. And what we're currently thinking is that let's go we're thinking of two things. Okay, so one thing is that this is short-term experiment, and we don't really know what happens when it's long-term. So if we are irrigating for a very long time and the soil or the bacteria in the soil are exposed for a very, very long time, so we don't know what happened then. And so we're trying to do two projects where we're trying to cast bioplastic films to see the effect of this bioplastics on antibiotic binding. And together with Clinton and Jonas and Meg, we're trying to increase the biodegradability maybe and use different mulch in the field. But as like a researcher, I don't get it. Okay, we're irrigating with E. coli. We're putting a lot of E. coli into the soil, and we're finding nothing. Doesn't make sense. Okay. We really want to understand what happens to so we're kind of saying, where have all the E. coli gone? What happened? And so soil is a very, very complex environment where you have not only bacteria, but you have a lot of other microorganisms. And so one hypothesis was that, the enteric bacteria that we're introducing with the treated wastewater are not detected, basically because they cannot compete with the indigenous bacteria within the soil. But another hypothesis was that actually the enteric bacteria that we are introducing with the treated wastewater are basically great food for the micropredators in the soil. So there are a lot of micropredators in the soil, nematodes, protists, predatory protists, other predatory bacteria. So maybe they're predating, they're kind of free food, nice, because we are introducing bacteria that have no resistance against these predators. Okay, bacteria that are coming with the water, and we're introducing them, and they cannot and they have no resistance they have no resistance to the predators in the soil. So the next thing we thought of, how are we going to prove that? Okay, so what experiment can we do? Simple experiment that can show that actually the bacteria are being predated in the soil. And so what we did was using antibiotics. It took us a while, But what we thought of doing is the following. Let's take soil, irrigate it with synthetic treated wastewater, add bacteria, and then add antibiotic that inhibits predators, in this case, protists, and see. So if it inhibits predators, we'll see the decrease in the E. coli would be less than the decrease without the antibiotic, right? Because the predators are basically inhibited. So we did the experiment, and we were very happy to see that it worked exactly as we anticipated. So with antibiotics, there was a 2 orders of magnitude less decrease. So the bacteria did not decrease as much as without antibiotics, which means that there are micropredators in the soil that are predating over the bacteria. And this is interesting both when you're using different types of fertilizers. Okay, so if you're using biosolids and we're afraid of the pathogen, maybe if we're using the right amount of pathogens and irrigating the soil, the right amount of irrigation, allowing connectivity in the soil such that the produce can move within the soil, it could increase predation, and then we could manage or mitigate contamination in the soil. So that was very interesting, and we were trying to do that in organic and conventional soil because we expected that in organic and conventional soil we'll find completely different communities. So this is an ongoing study that we're currently holding, trying to see which predators. Okay, so we know that there are predators that are predating over the enteric bacteria that we're introducing into the soil, but which are they, and what moisture level in the soil would accelerate this predation rather than, and how dry, if the soil is dry, would it inhibit predation? So we're trying to play with that. We're currently doing these experiments. And I'm not going to touch over the results of that. We're also trying to see whether the use of treated wastewater can inhibit the predation, okay? Because treated wastewater have higher concentration of salts, or the salinity is higher, and whether it's going to affect predation. We're also trying to see whether the fact that we're tilling the soil and we're changing the structure of the soil, would that affect predation? So we're trying to do these experiments in the lab and in the field. And, next time I can tell you about, the results of that. Okay, so our conclusions are that the pathogen diversity and the resistome, the entire antibiotic resistant bacteria in the soil are not correlated with the irrigation, with the water quality. I hope I convinced you, or the soil barriers that we're implementing, or the soil type. Okay, so they don't really care, and the presence and absence of antibiotic resistant genes were also not correlated with antibiotics or heavy metals or the environmental conditions or any of the other conditions that we applied. Though plastic mulch does make a difference. So it increases the antibiotics in the soil, and we hypothesize that over time it may increase antibiotic resistance. We also showed that fecal indicator are decreased in treated wastewater irrigated soil unless we inhibit the protists that are predating over them. And that their network analysis, we're showing that there are specific protists that are actually consuming these bacteria that we're introducing into the soil. And which protists they are and how they're exactly doing that, again, that's what we're currently doing. So a lot of people were participating, and a lot of institutions, people from different places, and the funding agencies are amazing for supporting our work. And thank you for your attention.